Hello everyone, I am Egle Cekanaviciute, a scientist in the Space Biosciences Research Branch at NASA Ames Research Center. And for this Plus 10 talk, I would like us to imagine how we could develop research on organs on a chip and organoid models to understand and mitigate the biological impact of the space environment on the human body, also known as Send My Brain to the Moon 2030. So why should we work on organ models for space exploration? Because they present an ideal model system by allowing us to investigate human responses to real and simulated spaceflight stressors, include complex interactions between different cell types in 3D systems. They also present a possibility to automatize the processes of cell growth, sampling, and sample analysis. And they are personalizable to quantify the health risk for a specific person and plan the countermeasures accordingly. And ideally, we could do all of the above in a sufficiently high throughput manner to maximize the amount of data collected. Currently, we already have organ models for all the main organ systems that are affected by spaceflight and deep space radiation. That would be central nervous system, cardiovascular system, immune, musculoskeletal system, cancer development, and host microbiome interactions. All of them are continuously developed and improved. This is just the So here is an example from our own work. This is a high-throughput human 3D neurovascular system based on using commercially available modified 384 well plates, which can contain up to 96 chips that are composed of multiple channels that could be seeded with cell types of interest to form 3D structures. Here is one of our chips with a model vessel in the top channel and an image of a different chip with different neurovascular cell types interacting with each other. So what could such chips be used for? We have already investigated how human neurovascular system reacts to space environment, specifically by modeling deep space radiation using iron particles, which are a major biologically active component of the galactic cosmic rays, and showed that they cause both cellular damage, with cells called astrocytes, depicted here, being particularly sensitive, and leakiness of the blood vessels, shown here using a fluorescent dye that is loaded in the top channel and leaks into the bottom channel. For leakiness, the same cells called astrocytes play a major role in blood vessel disruption. So this is how we have identified astrocytes as a potential weakest link for responding to deep space radiation and therefore forming a suitable target for countermeasures. But this is just the beginning. The real question is, how could we develop this and similar human organ models for the next 10 years? Well, first of all, we need to utilize the fact that these models can be seeded with cells that are derived from induced pluripotent stem cells, which in turn can be derived from a subject of interest. So if I want to go to Mars, and I want to go to Mars, I would like to see the risk profile and countermeasure responses in a model that is as close as possible to my own brain. In addition, such models could be used to compare sensitive and resistant individuals and uncover the pathways and mechanisms that are responsible for these individual differences, and could then be targeted to increase resistance in everyone. In addition, the high-throughput nature of organ models allows to use it for screening and validation of countermeasures. Our group, for example, is just beginning a collaborative project of drug repurposing from terrestrial to space medicine, using computational analysis to identify the overlapping issues and associated FDA-approved therapeutics. But all of them will then need to be validated experimentally in either simulated or real spaceflight conditions. And this can be done using the high-throughput human organ models. And finally, these models could be used to understand the responses to simulated spaceflight stressors such as microgravity and simulated deep space radiation, as well as eventual adaptation for payloads, especially because they lend themselves to high throughput and increased automatization compared to, for example, animal models. So in summary, this is why we need you to work on developing and improving human organ models that would go far beyond these first pass efforts that we are working on now, and eventually would make us able to model human health risks and develop human countermeasures for deep space exploration. Thank you very much for listening, and you can reach me using my email below.